sneaking around to get to my talk, and next year I'm going to cut it out, hopefully, because we'll have solved some of these issues and the questions. But we'll keep it quick. I usually love to do this talk. It's the what's next, because patients come and they go, well, I've seen all of that. What's next? What are you doing? You're saying that things are happening, but I'm seeing the same slides, and what are you doing? So I'm just going to tell you what's next. It's going to be quick and fun, and it just goes to show that at a time when I thought the immunotherapy is the end of it or targeted therapy is the end of it, it's just more than that. Here are my disclosures. I really like to make a point that if you came 3 years ago or 5 years ago, you'd see 3 names on there, but pharma is all in, and we have people from pharma here. I just want to take some time to acknowledge their work and their company's work in order to make this happen. The biomarker work, the work that allows us to understand what's going on in a time when the funding from the government is at an all-time low, comes from pharma. So thank you, and we need more of you on that list. Here's what was presented today. The combination targeted agents are doing well. The immunological agents are doing well. What we're doing is we're sort of bringing the survival out. We see a plateau, and it's wonderful, but we're not benefiting everyone. And at some point, we have to look at other avenues for care, and not only for melanoma. You people here are so important because you've shown that these therapies work not just for melanoma, but for all of these cancers. In a week where there was an approval for a drug for bladder cancer, I want you to know that that drug from Genentech, atezolizumab, which is called Tecentriq now, we opened at Angeles. We were the first place in the world to open, and we opened it for you, our melanoma patients. And from that, the data came that led to the approval for the bladder cancer patients. So they owe you all a debt, and we will collect. And this is what's happened, all of these approvals, even to May 18th. I pulled this off of Twitter because it's like up to the minute, all of these approvals, but we're moving forward because there must be more. And this was me and Jeff when we're at USC. Every once in a while they made us dress like Trojans. And that's Jeff when he had more hair, and this is Hamid when he had more hair. But we said there must be more. And we would go and tell these pharmaceutical companies, come on, give us more. And we have patients who need more. And these things are now here. They're in the clinic. They're available for everyone. And we all agree now that PD-1 is the backbone for the therapies that are moving forward, not just for any common melanoma, but for multiple solid tumors. And I pulled this up. I used to put this up, and everyone would gasp and say, wow, look at all these trials. But a review of what was on the internet just a month ago turned this into this. Okay? So this is an era of hope. It's an era of all in. It's an era of, looking to find the right view, and the view should be positive. And it's not just from the CTLA-4 and the PD-1, but immune modulatory receptors are coming out of the woodwork, and we have all of these, OX40, GITR, CD137, and there are these are the gas pedals that we're going to push on, and these are the brakes we're going to release, and these are all out there. Right now, I would have loved to introduce all of you to our second new physician, Sam Kleppner, but I sent him away, and he's away. He's coming back, though. He's away working with a pharmaceutical company that's brought OX40 in combination with CD137 to our clinic for our patients and our patients with melanoma. So this is now this. So there's more. CD40, and this is CD137, and all of these co-stimulators and these co-inhibitors. And, it's one gas pedal is not the same. There are tumors that preferentially express TIM3 or LAG3, and what we need to know is whether we should be giving this or this or this or this, and that's what we're trying to find out. And GITR is one of those stimulators. It's a member of the tumor necrosis. You don't need to remember any of this stuff. But what it is a stimulator to the immune response. And it's a development that we're looking at, and not just one. You know, when we were doing trials with ipilimumab, with Yervoy, we would beg for slots. They would say we're opening a trial for 50 patients, and Dr. Boasberg and I would say, okay, great, we'll take 45 of them. And they would say, no, it's opening in 5 places. And there was only one drug. And now there are like 4 or 5 different GITR antibodies and 8 different PD-1s, and everybody wants to work together to make this happen. GITR increases the inflammatory response. It leads to a more robust immune response. It helps to downregulate the FOXP3, the T-cell regulators. It does a lot, and it does a lot that the other stimulators don't. They have other roles that we're just beginning to learn about, and there is more to come. OX40 is another stepping on the gas, and it's a co-stimulatory molecule that promotes T-cell proliferation. It increases cytokines like everyone else does. But what we're finding is it enhances memory T-cell survival, those cells that stick around, that when you have another flare are ready to go. And these are being looked at. These are in clinical trials at my place and his place and her place and his place and all throughout the nation right now. So hopefully next year this won't be what's upcoming. It'll be part of the immune talk, and there'll be some great data. And the initial data is just being presented. And what we're learning is, these are mouse models, and I don't care much for mice except they begin to show us the promise of combining therapies. And these are the initial mouse models of the ipilimumab anti-CTLA-4 and the nivolumab anti-PD-1. And this is what convinced them to move ipi-nivo trials forward and brought us to the approval. And it just doesn't exist here. Here are other models of OX40 with 4-1BB, and these are in clinical trials. This is early data that shows that it works. It doesn't matter what this says. What it does say is it increases tumor T-cell infiltration is what we want. And in early trials, we're finding that combining two is tolerable and palatable. And our quest is to find something that works for people who have failed ipilimumab and nivolumab. That combo has failed to control their tumor. Maybe this combo can work. And we can also maybe find things that are less toxic. Here are two, a stimulator and a inhibitor, and we've seen the same things. The tumor volumes drop. But we're not just looking at the T cell. The microenvironment is important. And just quickly, I'll take you on a little voyage through here. Those of you who've heard about VEGF and Avastin, a drug that's approved and its immune roles, and this is being combined with PD-1 as are others. The future is in combination. IDO inhibitors are coming forward, and they work in the tumor microenvironment. They're required for states of immune suppression like pregnancy, but in cancer not necessary. Combinations with other cytokines, oncolytic viruses, which we haven't touched upon, I'll touch upon. This combo has been talked about multiple times. And yes, I'm here to tell you, everyone I brought here to talk is wrong. Chemotherapy does have a role. The role is fitting it into the appropriate places to control disease and to be a stimulator for a stress event that increases PD-L1 and allows the efficacy of these immunotherapies to increase. There's data in breast cancer that's showing that chemo and immuno can work. There's data in lung cancer, and we just need to look further to understand who needs it and who doesn't. The reason why we're not seeing benefit in all comers is because we're putting too many people on who don't need it. And these predictive markers that Dr. Sullivan talked about that we're sending him back to focus and work on are going to tell us not only who needs PD-1, who needs targeted therapy, but who can benefit from chemotherapy or from surgery. Next year, I don't want Dr. Essner saying that I'm hoity-toity. I want us to work well together. All right, so what we're doing is we're finding guidance. If there's a person with high PD-L1 in their tumor, we just need to release the brakes and block the checkpoint. We don't have enough PD-L1, we need to step on the gas, right? And if we don't have any gas and the car's not going, it's sitting on blocks in the front yard, we need to do more to fill the tank and generate the T cells. And this, while it seems like it is just a map without direction, is here. This is the answer. Oh, come back here. This is the answer. We have vaccines. We have radiation. Chemo. And we have anti-CTLA-4 to do this. And here are combinations that have been presented, and it's kind of stupid to present this and say what's coming, because this data is in our clinic and we're treating patients. I'm looking at someone who is being treated like this, and this is already gone from a phase 1 to a phase 2 to a randomized phase 3 that's being looked at in about 900 patients. IDO is epacadostat. IDO is upregulated around the tumor. It causes immune suppression. And we've looked at it in a phase 1 study in multiple different solid tumors, melanoma, lung cancer. This is the bladder patients, again, who owe us a debt of gratitude. The head and neck, the breast cancers, the ovarians. And what you've seen here is early data in melanoma looking at a response rate of 53%. Well, what does that mean? Well, if you paid attention to Dr. Sullivan talking about it being Evo, that response rate was about 57%. And these are very few toxicities were seen. There wasn't an increase in grade 3 toxicity. So this is a combination that's working, and we're looking to see if it can work for all comers. Again, tumor-associated macrophages promote tumor progression by increasing invasion. Angiogenesis, those are vessels that come and feed the tumor. They secrete immune suppressive factors, and they secrete factors to stimulate the growth and the motility of tumors from one place to another. And Dr. Carvajal and I are currently on a clinical trial where the goal is to accrue as many patients as possible. Actually, the goal is to accrue more patients than Carvajal. But to look at how this drug works in patients with melanoma and multiple solid tumors. Going quickly to oncolytic therapy with TVEC, which was just approved by the FDA, a local therapy that's injected into tumors. I like to say that this is like Mick Jagger in a hotel room, which you inject. This is a herpetic virus that's injected into the tumor. It preferentially gets picked up into the tumor, and just like Mick Jagger and the Stones, when they get into a hotel room, they multiply. I won't spend much time on that, and then they destroy the room, and there's a little joke about herpes somewhere in there, and then they move on to another room, and what they do is this tumor also codes for GM-CSF, which causes an immune stimulatory environment. Those immature dendritic cells, which are like the maids that come in and go, oh my God, look what Mick Jagger did to this room. They pick up the tumor fragments, and they mature. They go to T cells, and that's like the T cells we use to combat other tumors and cancer. And then those T cells can go to other areas in the body. And Imlygic, which is TVEC, this oncolytic immunotherapy has been approved. And this is what it does to local tumor. It sort of controls everything here so you don't have tumors growing. I want to point out that this was injected, these areas were injected, but not everywhere was injected. So there's a local effect. We can also see that the T cells go up after TVEC and after adding other immunotherapies. So this is a therapy that's been combined with ipilimumab and is being combined with pembrolizumab, and we're seeing higher response rate than any single agent alone and less toxicity than other combinations. And now it is going into a trial that's at many of our centers looking at TVEC and pembrolizumab versus placebo and pembrolizumab alone. So here's the proof. You inject this tumor, it shrinks and goes away. Here's another patient that had tumor injected, but a distant disease has resolved on its own based on being stimulated locally. I thought I'd bring this, and this would be my showstopper, but of course, when you have something that's so good, most people pick it up, and Dr. Carvajal stole it and presented it, and so did Dr. Ferries. But IMC-GP100 is a drug, and you don't have this slide, I'll send it to you. I call it the matchmaker or the Yenta. It is an antibody on each side, and what it does is it brings the T cell and the tumor and says, hey, you two get together. You guys got to get together. And just brings them together and creates this optimized immune system, which it just means it brings it together. The T cell gets activated without needing the normal ways it gets activated. So if you have someone whose T cells are not strong enough to get there on its own, you have a shy T cell, it makes it more, and it brings it in and it can kill the tumor. And this was the data that Mark Middleton presented at AACR, and these are the spider plots as we talk about it. 0 is the tumor bulk at beginning, and you want those tumors to shrink, not go higher. And again, this is early data, but you can see here, this is an ocular melanoma that went to a complete response for a time, and that's important. If it can work in ocular melanoma, it should work in other types of tumor. And we're not just stopping there. You know, I saved this last slide because I set everyone up here to talk about combination, single agent combination, and triple therapy. And then I'd come and I'd say, ha-ha, here is triple therapy that isn't just BRAF and MEK. Here it is. We're putting this drug together with anti-CTLA-4 and anti-PD-1. And so this is a new time, triple immunotherapy. And you can see you can throw more in. And we're going from a time of just having one therapy, one drug, to 2 to 3 to 4, and getting bigger and better combinations going. We talked about adoptive T-cell therapy, but we're working to make this better for everyone. And this is a trial where the patients don't need to go to a place that has a lab to grow the T-cells. We can cut it out, send the tumor to a central place, and then every clinic can do adoptive T-cell therapy. And that's the future, and that's coming. Rick, this is the drug that you introduced me to. And antibody-drug conjugates didn't start in melanoma. They're approved for breast cancer, and it's just a linker with a chemotherapeutic attached. And you can target anything you want, any protein. GPNMB is one. And we are currently doing a trial of CDX-011, which is an antibody-drug conjugate. There's a chemotherapeutic attached to an antibody. It's like a smart bomb. And that goes, I call it a Molotov cocktail mixed with a Trojan horse, which is hard for a Middle Eastern doctor to say. But it clamps on to the tumor, it gets internalized, and then the chemotherapy breaks apart, preferentially getting to the tumor. And when we just finished accruing 80 patients to this study as a single agent, and this data will be presented in Europe upcoming. But this trial is now changing, and it's going to be the combination of this antibody-drug conjugate. Here we go. This antibody-drug conjugate and an immuno-targeted agent. And we'll put another 60 patients on and see what happens. I did say that I wanted to surpass everyone, and there are trials now not looking at just 1 or 2 or 3, and this is a 4-agent trial that's coming through the NCI, where you get a little bit of BRAF and MEK, and then you go on to combo anti-CTLA-4 and anti-PD-1 together. So you'll see a month of targeted followed by continual immuno to see if we can do better. Dabrafenib, trametinib, ipilimumab, nivolumab. four things together. Weber talked a little bit about ERK inhibitors coming forward, and that might be the fifth agent that we put in. Who knows? The logic trial was presented already, but that's just not it. We have to do better, and we are. When we control systemic disease, places that tumors go to because they're so smart can be the brain. And this is the final frontier for me because it's so important. We're having patients come in with multiple different types of tumors, and we've done so well in increasing how well these patients do that the tumor then goes to a place where we don't want it. This is a patient that belongs to a guy whose name we haven't brought up much, but it's important, Steve Hody, who is a great contributor. This is a brain metastasis. This is your brain. This is your brain on ipilimumab. And this patient has not had radiation and the toxicities that may come with it. This patient has not had neurosurgery. I think it's okay if we talk ill about the neurosurgeons since they're not here. But just the treatment alone with ipilimumab has caused that to resolve and go away and stay away. And it's the same here with this Rick Keffert's patient on pembrolizumab, and that went away. And we're currently doing combination ipi-nivo, and we're seeing good results in the brain, and this will be presented shortly. One of the main points of Dr. Essner's talk was prevention, not just prevention of a initial de novo melanoma, but prevention of having patients who show up with a stage 2 and a stage 3 melanoma for them to have metastatic disease. And that's a major goal for our collaborations together, is to find that answer. Because adjuvant therapy, the therapy when we get rid of local disease and there is no metastases, is a gold standard in breast cancer, lung cancer, colorectal cancer. But in melanoma, we have not quite found the answers, and we are making the steps. We had not had great metastatic therapy, which we do now. And this BRAF and MEK targeted has been looked in the adjuvant. The data is coming. The best data that we have right now is ipilimumab in the adjuvant setting, which is showing a relapse-free survival. So it delays the time that this tumors come back. And hopefully in the upcoming year or year and a half, we'll have some overall survival data. We are looking for PD-1 because we believe that it can be less toxic than anti-CTLA-4 Yervoy therapy, a higher response rate, and possibly going to combination. And currently, Cedars Angeles collaboration and other centers are looking at pembrolizumab versus ipilimumab or interferon in an adjuvant trial. So our vision is prevent it, vision is develop drugs, and also this idea of a personalized program to figure out, is this an inflamed patient or non-inflamed patient? Is there immune, is it strong PD-L1? So maybe you just need a PD-1 inhibitor. Weak, maybe you might need that anti-CSF1R that Carvajal and I are working on too. Or maybe you need that IDO inhibitor, epacadostat. Maybe we need to increase the PD-L1. There's chemo again. There's Avastin, the VEGF to bring the T cells in. This bispecific is IMC GP100, which we talked about. We talked about OX40, and there's chemo again. So whereas if you put this up for me 5 years ago or 3 years ago, I would have said, all right, I got to go knock on this guy's door and this guy's door and this guy's door and say, hurry up. This is all here, and we will have better answers soon. I think it's great to say that we've made significant progress, but the best is yet to come. And with that, I want to thank you for joining us here. We'll just take a little minute to pick up your questions. Just put your question cards up, and I'll ask the